In the summer of 1872, a fossil hunter working a quarry outside Caen in northern France split open a slab of Jurassic limestone and found the skull of a crocodile inside it. The snout was long and narrow, the teeth interlocking, the skull architecture almost identical to that of a living gharial from the Ganges River. The skull was 160 million years old. The man who found it could be forgiven for assuming the animal had barely changed. That assumption would hold for 150 years. It would take a genome to break it. The crocodile became science's most familiar symbol of evolutionary stasis. It had survived the asteroid impact that killed the non-avian dinosaurs. It had survived the Permian extinction, the Triassic extinction, the Cretaceous extinction. It had watched continents drift and sea levels rise and fall, while other lineages vanished around it. Every museum display said the same thing. Crocodilians are living fossils, ancient survivors unchanged since the age of the dinosaurs. Every documentary repeated it. The image was too compelling to question. And almost every part of it was wrong. When molecular biologists finally read the crocodilian genome in detail, what they found was not an animal frozen in time. It was an animal that had been quietly and radically reshaping itself for 95 million years, building one of the most sophisticated biological architectures in vertebrate history, while its skeleton stayed the same. The first crack appeared not in a laboratory, but in a taxonomy dispute that had been running for decades. The gharial, Gavialis gangeticus, is one of the strangest-looking animals alive. It grows to over 5 meters. Its snout is extraordinarily elongated, covered in sensory organs, perfectly adapted for catching fish in fast-moving rivers. For most of the 20th century, paleontologists classified it as the most basal living crocodilian, the closest thing to the ancestral form, a survivor that had barely diverged from the original blueprint. The long, delicate snout was read as primitive, ancient, pre-modification. The gharial was used as a window into what all crocodilians had looked like before evolution pushed the others in different directions. In 2014, a team led by Richard Green at the University of California, Santa Cruz published the first high-quality genomes of all 3 living crocodilian lineages in the journal Science: the American alligator, the gharial, and the saltwater crocodile. The project had taken years. The results dismantled the old tree entirely. The gharial was not the most basal lineage. The gharial was not primitive. Phylogenetically, the gharial sat as the sister group of the true crocodiles, Crocodilus, meaning it had diverged relatively recently from the same branch that produced the saltwater, Nile, and American crocodiles. The long snout that everyone had read as ancient was a derived feature, an adaptation, not an inheritance. The gharial had grown that snout because catching fish in fast Himalayan rivers required it, not because its ancestors had always looked that way. The implications spread immediately across the entire field. A separate long-snouted form, the false gharial of Southeast Asia, Tomistoma schlegelii, had been classified alongside the Indian gharial for over 150 years purely on the basis of jaw shape. Long snout meant gharial lineage. That was the logic. The DNA ended it. The false gharial was not related to the Indian gharial. It sat firmly within the true crocodile branch. 2 animals that looked almost identical, that shared the same hydrodynamic jaw design, that lived thousands of kilometers apart, had arrived at nearly the same skull architecture through completely independent evolutionary paths. Long snouts had evolved at least 3 separate times in crocodilian history. The shape had been reinvented again and again because the physics of underwater fish catching kept producing the same optimal solution. The similarity that had misled taxonomy for a century and a half was not ancestry. it was physics. But the genome rearrangement was only the beginning of what the molecular data had to say. The Nile crocodile had been treated as a single species since Linnaeus named it in 1758. It ranges across sub-Saharan Africa, down to South Africa, and up into the Nile Delta. For 2.5 centuries, the enormous crocodile pulling wildebeest into the Mara River and the smaller, darker animal hunting along the West African coast were considered the same animal with regional variation. Population genetic studies published through the 2000s and early 2010s produced a different picture. The two populations had been genetically isolated for somewhere between 6 and 8 million years. Their mitochondrial DNA divergence was comparable to the gap between two recognized sister species. Researchers formally split them: Crocodilus niloticus in the east, Crocodilus sucus in the west, and along the Nile's upper reaches. The single Nile crocodile, familiar from every nature documentary, had never existed as a single thing. What looked like one animal from the outside had two separate evolutionary histories running for millions of years inside. The western species, Crocodilus sucus, turned out to carry a second secret. A 2011 genetic study led by Christopher Brochu at the University of Iowa compared it against crocodile species across the Atlantic. The American crocodile, Crocodilus acutus, the only crocodilian native to both North and South America, turned out to be most closely related not to any other New World species, but to the Western Nile crocodile lineage of Africa. The divergence time matched a window when sea levels and current patterns in the Atlantic would have made long-distance oceanic dispersal possible. Crocodiles, it appeared, had crossed an ocean. A population of West African crocodiles had drifted across the Atlantic, almost certainly on rafts of vegetation, sometime in the last 5 to 6 million years, colonized the Americas, and evolved in isolation into a species that taxonomists had always assumed had deep New World roots. The American crocodile was not native to the Americas in any deep sense. It was an African immigrant that arrived before humans, before the land bridges, before anything that left a written record. The Green Team's genome paper contained one more finding that was stranger than any of the rest. When they measured the rate at which the crocodilian genome was accumulating mutations over time, the number they got was almost impossible to believe. Crocodilian genomes evolve at approximately 1/5 the rate of mammal genomes, possibly slower. The molecular clock that ticks through every living lineage, accumulating random mutations at a roughly predictable rate across generations, was nearly stopped in crocodilians. They are the most evolutionarily conservative vertebrates on Earth, with a genome that changes so slowly it almost reads as geological rather than biological. This was not what anyone expected to find in an animal that had survived 5 mass extinctions. The standard assumption was that survival required flexibility, rapid adaptation, the ability to generate variation fast. The crocodilian genome suggested a different model entirely. What it showed instead was extreme genomic stability, paired with extraordinary physiological complexity. The immune system genes in the crocodilian genome are among the most elaborate in any vertebrate, built to handle the bacterial load of carrion feeding, deep puncture wounds, and sitting in warm, contaminated water for months at a time. The cardiovascular system, which allows crocodilians to shut blood flow to non-essential organs and sustain extended dives, is encoded in the genome with a level of detail that still has cardiologists paying attention. The metabolic genes allow a crocodile to survive a year without food, digest bone, and then sprint at 17 kilometers per hour when it needs to. None of these capabilities were visible from the outside. All of them were written in the sequence that everyone had assumed was essentially unchanged since the Mesozoic. The living fossil story was always too simple. The skeleton stayed recognizable. The genome did not. What the DNA revealed in crocodilians was not a lineage that had stopped evolving, but a lineage that had evolved differently from everything else, channeling its change into chemistry and physiology rather than shape and form. The result was an animal that could spend 95 million years looking like a crocodile while rebuilding its biology from the inside out, surviving everything the planet threw at it, not through luck or stasis, but through a level of internal engineering that the fossil record had no way to show. The gharial's snout was not ancient. The Nile crocodile was not one animal. The American crocodile was not American. And the genome that produced all of this was the slowest changing invertebrate life, accumulating its innovations in a register that bones and scales and skulls could never record. Every crocodile lying motionless at the water's edge carries a history that the surface gives away nothing about. The DNA finally told what that history actually was. If this story changed how you see a crocodile, there are dozens more waiting on this channel. Every lineage has a hidden genetic history. Subscribe, and you will not miss the next one.